All right, hi everyone, and welcome to the Wednesday Compass Seminar. I am super excited to introduce Liz. Last name?
Yanuskiewicz.
Liz is a fourth-year PhD candidate working with Professor Close in the Ocean Sciences Department, researching major components and alteration processes of marine particulate organic matter. Before joining us at the Rosenstiel School, she received her master's from Eckerd College in 2016 and a master's from the University of North Carolina, Wilmington in 2019. Both of these degrees were in marine science. Before starting her PhD, she was a laboratory technician with, I believe, Professor Close. And even though she didn't say this in the email, she was also a park ranger, which is like a pretty cool job, I would say.
That's pretty cool.
For the Forest Service. Outside of research, Liz is well known for playing rugby. And she is also going to be defending her dissertation this summer, so make sure to come by for that as well. So if we could all welcome Liz.
We don't show the abstract for hours. Do you show the abstract?
No.
Okay, cool. Are we recording? Okay, great. Awesome. All right. Well, thanks for coming. So today I'm going to chat about the sources and transformation of particulate organic matter in the North Atlantic spring bloom. And so the research that I'll share with you today is part of my chapters 1 and 2. So the manuscripts are in preparation. And then, like James said, I am planning a summer defense. So if you want to hear about my chapter 3, come by then. So I just wanted to start off with what is marine particulate organic matter, or at least how do we define it. And so if we imagine a liter of seawater, there's a lot of stuff in there, such as viruses, fecal pellets, microplastics, zooplankton, and phytoplankton, just to name a few. But we're interested in organic matter. So any anthropogenically derived material like microplastics we exclude during our quality control processing. And then organic or zooplankton are organic matter, but they have the ability to swim. So we also exclude them from our definition because we're interested in what is passively settling through the water column in the particulate form. And we can collect that on filters just like this. So why do we care about particulate organic matter? Well, if we look at the near surface, a lot of the components that we just talked about on the last slide, like phytoplankton, microbes, fecal pellets, they make up the base of the marine food web. And then particles also dominate the export of organic carbon. And an example of why this is important, so if we have particles that form large aggregates, they can sink more quickly to depth and even reach the seafloor. And this is important for transferring organic carbon into the ocean interior where it cannot be readily exchanged with the atmosphere. But then there's also particles that are smaller. They may not be acquired into aggregates. And so the transformation and the export of small particles may be different between the different sizes. So it's also important to recognize how organic matter may be transformed throughout the water column and what the different sizes may implicate. And if we look at the sources and the transformations of particulate organic matter in the water column, this can tell us about information about all of these sources that are impacting particle export. And then with this knowledge, we can validate global carbon export models, which is urgently needed if we are to accurately predict global carbon export in future climate scenarios. So one example of why it's important to understand the processes that control particle export. So all of these maps are global carbon export models, and these maps are representing the fraction of net primary production that is exported. And all of these models are slightly different, so they have different inputs. And based on what you are feeding the model, we can see that there's quite a lot of variability. And so this makes predicting global carbon export a challenge. But an approach towards mitigating that challenge is to synthesize in situ data with satellite observations to improve these models. And this is the approach that the export processes in the Ocean from Remote Sensing team is doing. And so the research that I'll be showing you today is part of the exports project. And so I've been working with particles or marine particulate organic matter in the Northeast Atlantic. So the exports team sampled this site in May of 2021 during a declining spring phytoplankton bloom. And throughout my talk, there will be a portion where I compare the Northeast Atlantic particles to that of the particle data set analyzed by Voital et al., who looked at particles in the Northeast Pacific. And this was export's first field site. And it's really great that we have this comparison because we can see, looking at this, this is another model output of the fraction of net primary production that is exported. And if we look at these estimates from this model, we see that in the Atlantic, it's expected to have greater carbon export in comparison to the Pacific. So we have this great opportunity to compare particle data sets and look at the sources and transformations in both water columns. So, how were the particles collected? The exports team utilized a variety of research vessels, and on one of those research vessels, there were these large volume in situ McLean pumps. And so the pumps are down here at the bottom of the metal frame. And then at the top, we have these filter holders. And inside the filter holders, we had sequentially stacked filters. And this is probably going to be loud, so hold on. Oh, I muted it. Okay. Sorry, Zoom, if it's loud. But once the pumps were programmed and the filters were loaded, we hauled the entire pump array over the side of the ship, clipped it onto the wire, and it went down to a predetermined depth, where it pumped for several hours in order to collect enough material for us to do our chemical analysis. And so in the Atlantic, we had 5 different sizes of particles collected. We had 2 that are in the purple that are small particles, and then we had 3 which we term large particles. Sediment traps were also deployed. And so just to simplify throughout the rest of this talk, I'll be grouping particles into small particles, large particles, and sinking particles. And if I call out a feature specifically within one of these size classes, it'll be evident. And so in our small particles, we expect to find signatures of bacteria. And in our large particles, we expect diatoms, aggregates, and fecal pellets to be a part of the large and sinking particles. And as I mentioned earlier on the second slide about the different, the aggregate sinking versus the small particle sinking, it's important to recognize that the difference in sizes of these particles relates to different sinking rates. So then this leads to different sequestration potential. And also these different sizes of particles can be used differently by the food web, and they're also subject to different alteration processes. So now I just want to orient everybody to the water column that was sampled in the Northeast Atlantic exports campaign. And first off, the exports campaign sampled a retentive anticyclonic eddy. And the idea behind this was mainly to try to minimize physical advection, which for the most part was successful, except for waters deeper than about 100 meters. But however, these surface eddy core waters were exchanged with outside waters that did not compromise the eddy. And this mainly happened because there were storm events. So we had 4 major storm events that exchanged those surface waters and also deepened the mixed layer. And these storm events also bracketed the 3 dates we have particles. So we collected particles on May 13th, the 21st, and the 27th. And so you may hear me say throughout the talk the early time point and the late time point. So when I mean early, I mean this time point, May 13th, and then the latest time point is May 27th. And so we collected particles during a declining spring phytoplankton bloom. When the exports team arrived on site, silicate levels were low, which indicated that the diatom bloom already peaked and we were sampling a declining spring phytoplankton bloom. And so we collected small and large particles in the euphotic zone at these 3 time points. And the euphotic zone is the sunlit layer of the water column. So this is where primary production is happening, where a lot of our particles are being produced. But then there's another layer of the water column, the upper mesopelagic. And in this region, and particularly directly below the euphotic zone, particles are rapidly remineralized. And so we wanted to collect particles in this region of the water column to really look at the transformations happening to the different sizes of particles. And then also looking at the particles that are in this layer of the water column, this can tell us about how much material is actually being exported out of the surface ocean. And then we also collected particles in the mesopelagic. And these particles can tell us a little bit about the transfer efficiency. So there's the standard definition that once you're 100 meters below the euphotic zone, this can give you an idea of how efficiently your particle is being exported. And so these particles down here in the mesopelagic are representative of particles that persisted through remineralization through the water column. And just one thing I really want to make evident is the whole point of this slide and animation was to really just show you where in the water column we sampled, but I did not scale these shapes to size so they don't reflect quantities or concentrations, because in fact, in the euphotic zone, from the euphotic zone to the mesopelagic, we lost about 95% of the particulate organic matter. So now that we have our background, how do we determine the sources and the transformation that's affecting these particles during export? Well, we targeted amino acids and carbohydrates because together they make up a large proportion of the particulate organic matter in the water column. And we can verify this with this figure from Lee et al. So if we look at the top bar plot, which is representative of plankton, we see that of the identifiable particulate organic carbon, the majority of it is carbohydrates and amino acids. And the same holds true for bacteria. So we'll first focus on amino acids. So amino acids are the building blocks of proteins, and just like humans, we need them for metabolism and cell growth. So do other organisms in the marine environment. And so we can use the patterns of the delta N15 values of the amino acids because these reflect source, metazoan, microbial signatures that contribute to and transform particles. And before I show you data of the nitrogen isotopes of the amino acids, I just want to walk through our notation. So this delta N15 notation represents the ratio of nitrogen 15 to nitrogen 14 of our sample in relation to the nitrogen 15 to nitrogen 14 of the standard in the delta notation. So you'll hear me say delta N15 a lot in the next few slides. And the reason we can use the nitrogen isotope patterns of amino acids is because the isotopes have different masses, so therefore there's different kinetic reaction rates, which leads to isotope fractionation. And an example of this is if we have our reactant pool and a reaction is happening, our lighter isotope will proceed at a faster rate, more so than the heavier isotope. So our product will be enriched in our lighter isotope, and therefore it will be depleted in the delta N15 signature in comparison to the reactant. So now we have to extract the amino acids from our particles. So this is what one of our filters looks like after it's been dried. And I took a split of this filter for amino acids. We have to do an acid hydrolysis, essentially to release smaller protein and peptide molecules from larger proteins that we can't analytically analyze. And so this just represents the smaller cut up pieces of our filters with some acid. They undergo a reaction. We have to remove the filter pulp, and then we go through a few purification steps. So a lot of wet chemistry, and ultimately you end up in a small volume of amino acids, which you can see this sample actually has a little bit of color in it. And then we get to analyze them in-house. So we use a gas chromatograph combustion isotope ratio mass spectrometer. And then with some data processing, we arrive at our delta N15 amino acid values. So now that we have our values, we can look at the patterns and try to understand the sources and transformations of particles happening in the North Atlantic. So we'll first look at the delta N15 signatures of our source amino acid values. And this parameter is helpful because if we see positive shifts in the delta N15 of the source values, this indicates that microbial protein hydrolysis occurred. And what I mean by microbial protein hydrolysis, so say we have our particle pool and there's bacteria present. Bacteria typically have ectoenzymes. We'll talk about polymers later. So they have ectoenzymes that work to degrade particles into smaller units that the bacteria can utilize. And so through this reaction, the particles or the organic matter that the bacteria degrade for their own use will be enriched in nitrogen-14, and then the particles that are left are nitrogen-15 enriched. And so this signature will show up in the delta N15 source values. And so what we saw in the Atlantic, we saw microbial protein hydrolysis activity on small particles, but not larger sinking particles. And so when we look at our particles, we have our delta N15 of our source values on the x-axis, and then we have increasing depth on the y-axis for our small, large, and sinking particles across all three sample time points, which are representative of the blue hues. And so we see that increasing delta N15 source value with depth for our small particles, but this feature is largely absent in the large and sinking particles. So we see this microbial protein hydrolysis acting on small particles, but not really larger sinking particles. Another useful parameter we can use is the trophic position. So in particles, if you see an increased trophic position, this indicates there's heterotrophic biomass or waste added to the particle pool. And what we saw in the Atlantic was the addition of heterotrophic biomass to small particles with depth. And then we saw a temporal addition of heterotrophic biomass or waste to our large and sinking particles. And so we have our trophic position on our x-axis with increasing depth on the y, once again for small, large, and sinking particles across all time points. And again, we see a similar feature to what we saw in the last slide. The small particles have this increasing trophic position with depth, indicating that there's addition of heterotrophic biomass or waste. And we don't really see that in the large or sinking particles, but we did notice that there are these temporal offsets. So in our early time point, our May 13th, which is our lightest blue, in the mesopelagic, they have a higher trophic position in large and sinking particles, which suggests that there may have been a more active food web happening earlier on, which was transforming our particles in comparison to our later time point. The third useful parameter that we can use when we look at the nitrogen isotope values of amino acids is if we specifically look at the amino acid threonine. So this amino acid is useful because when there's a negative shift in the delta N15 threonine value, this indicates that animal metabolic processing of POM occurred, and this typically shows up in fecal pellets in our particles. And so what we saw in the Atlantic was increased contribution of disaggregated fecal pellets to small particles with depth, and then we once again saw a temporal trend in our larger sinking particles. So we have our threonine values, and then with depth in our small particles, we have this decreasing delta N15 threonine value indicative of metabolic processing, so disaggregated fecal pellets were added to the small particle pool. And the large and sinking particles remain relatively invariant with increasing depth, but there is a temporal trend again. So this, together with the temporal trend in the trophic position, once again suggests that there was a more active food web transforming the large and sinking particles earlier on in the sampling campaign in comparison to later. And so the last one I'll show you is this term called SUM-V, and it's a calculation using various delta N15 values of amino acids. And the way we interpret SUM-V is if we see an increase in this parameter, this is progressive resynthesis of amino acids by bacteria. And so what we saw in the Atlantic, we saw heterotrophic resynthesis of amino acids in small particles, but once again, not in large or sinking particles. So when we look at the data, we see that in small particles, the SUM-V parameter is increasing with increasing depth, and we do not really see that in the larger sinking particles. So now we'll compare both export sites. So we have the high productivity site in the North Atlantic, and we'll compare that to the work done by Voital et al. In the Northeast Pacific, and we'll compare those 4 parameters that we just walked through. And so we'll start with small particles first. And overall, we saw a similar pattern between both export sites, even though there are different productivity regimes, but there was greater microbial activity that transformed smaller particles in the Pacific in comparison to the Atlantic. So because both export sites had different time points, I've just simplified this and took averages. And so we have our 4 parameters that we just walked through on the x-axis, and then we have our increasing depth on the y. The blue symbols are representative of the Atlantic, and then the orange is that of the Pacific from Voital et al. And when we just look at these patterns, we really do see the similar patterns. Wow, you cannot see that on here. But we see similar patterns happening in all depth profiles. But you will notice that in the delta N15 source values and the SUM-V, there is a greater change with depth in the Pacific. So now we'll do the same but for large particles. And what we saw when we did the large particle comparison is that there was digestive processing of particulate organic matter, and this was a major mechanism that transformed large particles with depth in the Pacific, and this showed up as fecal pellets. So we have those same parameters on the x-axis, increasing depth on the y, and now we're looking at our large particles. And when we look at the patterns with depth, we see that the Pacific particles have a greater change with these parameters with depth in comparison to the Atlantic. So now we just walked through some of the major ways particles are transformed throughout the water column at both exports locations, but we can take this step, we can take this research a step further, and we can also quantify the particulate organic matter components using an isotopic classification method that was proposed by a former lab member, Shannon Dordery. And so what Shannon did was she determined that the delta N15 values of the amino acids alanine and threonine normalized to local nitrogen isotope baseline values, could distinguish between fecal pellets, microbially degraded organic matter, phytoplankton biomass from zooplankton biomass. And this is great because, as I mentioned on the first slide, we do not consider zooplankton biomass as passively settling particles. And so we expect these three end members to contribute to our particles, with particles that fall closer to the phytoplankton end member representing fresher material, and then particles that are closer to the other two end members representative of more degraded material. So now I'll show you this same 2D space with our established end members, and I'm going to plot the Atlantic and Pacific particle data on top. And when we did this, we saw that overall there was less progression of degradation in the Atlantic particles. And so to prove that, we'll plot the data with our established end members. And this is the data from Voital et al. First. And so this is all the particle data. The different shapes represent the different classes of particles. And then the size represents the depth. So the larger shapes represent particles collected at surface waters, and then these particles were collected deeper. And so what we see is that the shallower particles are fresher. They fall closer to the phytoplankton end member. And then as we move along this with increasing depth, the particles are becoming more degraded. And so now we can plot the Atlantic particles over top. And there's a lot going on, but we see the same trend, that shallower particles are fresher, and then with depth, they're becoming more degraded. And so another really cool thing we can use with this tool is we can then ask ourselves the question, well, what is the composition of sinking particles? And the reason we'll focus on sinking particles is because these are the particles that are modeled in global carbon export models. So we'll take the delta N15 values of these amino acids and we'll construct a mixing model to get estimates of the phytoplankton, fecal pellet, and microbially degraded organic matter that is contributing to the sinking particles in both the Pacific and in the Atlantic. And so when we do that, spoiler alert, there's way more. What we see is a significant microbially degraded signature in the Atlantic that's not present in the Pacific, and this pathway is also absent from models. So to show you that, we'll first look at the phytoplankton and the fecal pellet end member, with the horizontal lines just representing the average euphotic depth at both exports locations. And what Voital et al. Observed in the Pacific was this dominant fecal pellet flux. About 80% of the sinking particles were comprised of fecal pellet, and this agreed with food web models. And then if we plot the Atlantic particles, or sinking particles, we see that there is a greater phytoplankton flux in the Pacific in comparison to the Atlantic, or backwards. Greater phytoplankton flux in the Atlantic in comparison to the Pacific, and this also was in agreement with food web models. And typically food web models consider sinking particles to be either fecal pellet flux or phytoplankton, and if we were just considering these two end members, then we would expect that the fecal pellet flux in the Atlantic to be greater, similar to what was observed in the Pacific. But that's not what we saw, and that's because when we include this third end member in the Atlantic, we see this significant microbially degraded signature, which enforces the idea that in future global carbon export models, we should be considering this microbially degraded signature. And so now we're just going to focus on the Northeast Atlantic. So what we just saw from the amino acid-based model was that phytoplankton and microbially degraded organic matter were important components of sinking particles. And we wanted to ask ourselves the question of, in this environment, what other important components of particles are there, and what other biochemicals should we be considering? And so studies at this site have repeatedly shown high C to N ratios. And this is the ratio of carbon to nitrogen. So a high C to N ratio means that there's excess carbon in relationship to nitrogen. And we also observed this along with export's collaborators. So at 3 time points, we observed, just in the specific size class of particles, elevated C to N ratios. And so this made us suspect that carbohydrates are also important at this site. So we wanted to apply approaches that look at the sources and transformations of particles when considering the carbohydrate concentrations. And so as we saw earlier, carbohydrates comprise a significant fraction of particulate organic matter. And I may interchange carbohydrates with sugars, and I mean the same thing. But just like for humans, carbohydrates are our main fuel source, and it's a similar thing can be said for phytoplankton. Phytoplankton consume carbohydrates at night, and they also produce carbohydrates during the day because it's a primary photosynthetic product. And carbohydrates can exist in different chemical forms, so they can be single sugar units like monosaccharides, and they can also be multiple sugar units, which are polysaccharides. And so some carbohydrates are labile, like the glucose typically is considered a labile sugar, which therefore it undergoes degradation. It's easily accessible by other organisms or the organisms that produce it itself as an energy source. Whereas some carbohydrates are refractory. So these would more be your structural carbohydrates. They tend to persist in the water column more so than the labile carbohydrates. And so since they persist, they can tell us information about their source and the transformation. So before we can learn about the source and transformation of particles from looking at carbohydrates, we have to extract them from our particles. And it's a similar approach to the amino acids. We start with our freeze-dried filter. We take a smaller split because we're looking at carbon, which is much more abundant than nitrogen. We do a hydrolysis to release the complex and large carbohydrates into monomers that are analytically detected. We still have to remove the filter pulp, but ultimately we once again end up in a very small volume of our carbohydrates. And then this was not done in-house. I went to the University of California, Santa Barbara and used their HPLC system because they are already equipped to look at carbohydrates. It's not a very common analytical technique. So also thank you to Drs. Lillian Henderson and Chance English and the Carlson Lab who helped me do this. And so after it goes through the HPLC, we arrive at our carbohydrate concentrations after some data processing. And so the carbohydrates that we're able to measure with this method, we have neutral sugars that show up in cellular material or storage and structural material. There's also amino sugars, which show up in peptidoglycan or chitin, which is also structural. There's acidic sugars that we can think of them as like sticky, so they facilitate particle aggregation, or they're abundant in transparent exopolymer particles, or TEP, and also biofilms. And then deoxy sugars are another important carbohydrate group to consider because they also facilitate particle aggregation. So since phytoplankton are a main group of organisms that produce carbohydrates, it's also important to understand which phytoplankton were present during our sampling. And so early on, diatoms were the most abundant photoautotroph in the surface ocean, and they produce quite a variety of carbohydrates. And then with time, so we're moving from May 13th to May 27th, there was an increase in chlorophytes, haptophytes, and dinoflagellates. all of these autotrophs also produce a variety of carbohydrates. And based on cell size alone, I expect the chlorophytes to be present in our small particles, and then the diatoms, haptophytes, and dinoflagellates to be present in our large particles. And an example of why it's important to look at the community of who's there when considering carbohydrate concentrations is because we saw temporal trends. So early on, large phytoplankton were the ones that were performing a lot of the primary production. But then by the later time point, it switched, and then the small phytoplankton were the producers. And so early on, the species of diatom Catoceros was a major player for primary production, but it also is known to produce a lot of deoxy sugars, which facilitate particle aggregation. But then with time, its abundance in the surface ocean really decreased. And so this could have implications for carbon export if these diatoms are producing carbohydrates that facilitate particle aggregation, and then they're not anymore. So now we can look at the carbohydrate composition in the ocean at our site. And so this is a PCA plot. PCA is just a data reduction tool, and this is showing all size classes of particles in our three main depth zones, the euphotic, the upper mesopelagic, and the mesopelagic, for our 3 time points. And what we observed in the surface ocean at 30 meters was just this dominance of glucose. So regardless of particle size or time, our surface particles all have this dominance of glucose. And glucose is readily produced, but it's also readily consumed. And so as we transition deeper into the water column, glucose is consumed, and then we see this persistence of sugars associated with more structural polysaccharides like lactose and rhamnose. And my advisor, Hillary Close, just shared with me a really great way to think about this is if you're going to order peel and eat shrimp at a restaurant, you're not going to eat the carapace. You're going to take off the carapace, and you're going to eat the shrimp. And then if you just throw the carapace on the ground, that's going to persist. So it's kind of, it's I don't even know. I'm not going to go there. But that's the way to think about it. So we saw that glucose was dominant in the surface ocean, and it represents recent photosynthate products, and it also follows this productivity shift that we saw from large phytoplankton dominating productivity rates, and then we see this transition into the small phytoplankton being responsible for most of the production later on in the field campaign. And what I mean by glucose follows this productivity shift. So this is a bar plot of glucose as a percentage of POC at 30 meters. And so we have our 5 size classes of particles across our 3 time points. And we see that our 2 smallest particles, there's an increase in glucose as a percentage of POC with time. So glucose is being produced. Whereas in our large particles, glucose is decreasing, except for the largest particle. But for the most part, it is decreasing with time. And so this reflects that phylop shift that was observed during the exports campaign. And since glucose is representative of photosynthate products, I wanted to test the validity of the amino acid-based model. And while this is not the strongest relationship, the relationship is significant, but I just wanted to go back to something that I mentioned earlier on, that the amino acid model does a fairly okay job at predicting data in an environment like the North Atlantic where carbohydrates are abundant. So we talked about glucose in the surface ocean, Now we'll look at depth and temporal trends of carbohydrate compositions. So once again, we have our small particles where we expect bacterial signatures, and then we have our large particles where we expect aggregates, diatoms, and fecal pellets. And so we'll look for depth patterns. So we'll look at the composition of carbohydrates in the euphotic zone in comparison to the mesopelagic. We do have data from this middle depth, but just to simplify, in the interest of time, we're just going to look for the major differences in the euphotic and versus the mesopelagic. And then same for the time point. We do have a third time point, but I just really wanted to look at the biggest differences that we see early on in comparison to our late time point. And so this data that's coming up, you can't really see it here, sorry, but May 13th will be this lighter blue, and then May 27th is representative of this darker blue. And so besides glucose, what did the other carbohydrates look like in the euphotic zone? Overall, we saw polysaccharides associated with diatom cell walls for all particles. And an example of this is if we look at our largest size class of particles are greater than 335 micron particles. We see at both time points galactose is high in abundance, and galactose is a sugar that is associated with diatom cell walls, so a structural component of cell material. But then with time, we did see a temporal shift from deoxy sugars, which facilitate particle aggregation, to more structural sugars in particles. And an example of this is in our 6 to 51 micrometer particles. We see this decrease from May 13th to more deoxy sugars, which are thought to facilitate particle aggregation and therefore help facilitate carbon export. And then by May 27th, their abundance decreases. And so now we'll look in the mesopelagic. And overall, we saw those structural polysaccharides transferred to depth. And so an example of this, now I'm showing you our 51 to 335 micrometer particles. And once again, we see this abundance of galactose persisting down into the mesopelagic at 300 meters, which indicates that these structural polysaccharides were transferred to depth and resisted degradation. And then we also saw a temporal pattern. So there was a switch from polysaccharides that were derived from phytoplankton to polysaccharides that were derived from microbial origins. And so an example of that we can see in our largest particles. So on May 13th, we had galactouronic acid and glucosamine, which are common sugars found in acidic and amino sugars in diatoms, but in phytoplankton. And then by May 27th, the biggest mole percentage we see is this galactosamine, and this is thought to have microbial origins. So in our largest particles in the mesopelagic, by our later time point, we see polysaccharides of this microbial origin. In comparison to early on, they were derived from fresher phytoplankton polysaccharides. So that might have been a lot, but now we're going to put together the delta N15 patterns of the amino acids and put that together with the carbohydrate concentrations to look at the temporal and depth evolution of particles during the North Atlantic spring bloom. And once again, to just simplify it a little bit more, we're just going to consider the euphotic zone, the mesopelagic, and our early and latest time point. And so on May 13th, we saw that small particles looked like phytoplankton. By May 27th, they still looked like phytoplankton. However, our large particles that looked like phytoplankton originally underwent transformations and had more heterotrophic waste by our later time point. We also saw evidence of particle aggregate components early on, and this was less abundant at our later time point. With depth, at both time points, the small particles were transformed by hydrolysis and heterotrophic processing, whereas the large particles did not see those processes, but early on, they looked like phytoplankton aggregates with more fecal pellet contributions. And then our large particles later on actually looked fresher. So it was phytoplankton aggregates with less fecal pellet contributions. And then lastly, our polysaccharides at our early time point looked like they were derived from phytoplankton. And then by our later time point, they looked like they were from microbial origins. And one thing that I did not animate on the slide is the way I have it right now, it kind of just looks like what is in the euphotic zone was transformed and sank into the mesopelagic. But based on this large particle class, which looked fresher in comparison to our surface particles that had more heterotrophic waste, we think there's a temporal offset, and actually these fresher particles that we saw earlier on in the euphotic zone were transferred and detected later on in the mesopelagic. And so, because this is part of the exports project, we have a lot of collaborators, so we get to put all of our data together, and that's what I will attempt to do with this slide. So we once again see on May 13th, everything looks like phytoplankton, and we see particle aggregate components that were most likely derived from this phytoplankton, this diatom-rich community. However, there was a storm just before and after May 13th, which deepened the mixed layer. It transferred these large phytoplankton to depth, where we see these phytoplankton-derived polysaccharides persist. But as these large phytoplankton were exported below the euphotic zone, there was hydrolysis and heterotrophic processing, and exports collaborators noted an increase in microbial respiration, which is indicative of these little Pac-Man guys. And also, with this deepening of the mixed layer and the export of larger phytoplankton below the euphotic zone, there was also at this time an observation of an increase in zooplankton and particle encounter rates. So these zooplankton likely played an important role in disaggregating particles and adding fecal pellets to the particle pools, which we saw in our large particle with the fecal pellet abundance. And then so by May 27th, the phytoplankton phyla shifted to a more diverse community with haptophytes and dinoflagellates and chlorophytes. And experts collaborators noted this massive sedimentation event prior to May 27th. So the surface community was likely exported, and at this time the subeuphotic respiration rates were the highest they were, which also relates to the hydrolysis and the heterotrophic processing we saw in the small particles. But then we saw our large particles that were actually fresher with less fecal pellet aggregates with depth. And then the polysaccharides looked like they were from a microbial origin. And so we also think that with this temporal offset, these fresher phytoplankton that were in the euphotic zone earlier on were also captured in the sedimentation event and were reflected in our large particle signatures. And so I have like a minute or two left. So this is really satisfying that our particle data really coincide with what our other collaborators found with very different techniques. But what's really unique about this data set is the high resolution of size fractionation. So I just wanted to go through some unique features that we saw in our size fractionated particles that we otherwise would not see if we were just considering small and large particles. So specifically the 6 to 51 micrometer particles, we suggest represents the actively growing phytoplankton community. And so here's a plot again of our delta N15 amino acid values. And just what I want you to focus on is the 6 to 51 micrometer particle class, which we do consider a large particle, but the trophic position by our latest time point actually decreases, which would represent phytoplankton. When we look at the other larger particles, their trophic position increases, and we saw that heterotrophic biomass or waste contribution to those larger particles. So we suggest that this size class represents the actively growing phytoplankton community. And if you remember from way early on, this is that same size class that has those elevated C to N ratios. And lastly, the size class of particles also showed that shift in the near surface phytoplankton community that was observed, but it's really cool that we got to see it come out in the chemical features. So this is another PCA space. This is an approach, well, I don't have the label. It's the delta C13 fingerprinting approach. And so this is the now carbon isotope ratios of the amino acids of our particles in comparison to established end members from the literature. And the big idea behind this is that when you plot your data on top of these established end members, we can hopefully get a fingerprint of which autotrophs are synthesizing those amino acids. And when we look just at the 6 to 51 micrometer particles, we see that on May 13th, so this diamond right here in the light blue, it looks like a diatom. And that fits in with what we saw from our observations and exports observations. But then by May 27th, we see this shift into the dinoflagellate and haptophyte region, which also agrees with what exports observed during the sampling campaign. So just a big take-home message from this work is that the uniqueness really falls in this high-resolution size fractionation, where we can really pick out different things happening in the particles, and then this allows us to understand the transformations happening to the different particle pools across different regimes, which can hopefully improve global carbon export models. And with that, I would just like to thank everyone on the screen. Thank you guys for listening to Particles. I'll take any questions.
Anyone in the room have questions?
Great talk, loved it, especially because it's a matter that I don't really know much about. For the second portion of the presentation, if you had 3 data collection days, why did you only focus on the early and the end instead of looking at the middle as well?
That's where we saw the biggest changes, but also a big part of it, the second portion, we're really tying it into what our collaborators are doing with zooplankton. And so the zooplankton, that group is trying to look at the sources, the food sources for the zooplankton. And so the particles stand a good chance at being those food sources. And they had it the early and the late time point. So I just wanted to match them, honestly. But I do have that middle point, you're right. And that middle point's actually really interesting because that's when we see the deepest, there was a big storm and the mixed layer reached its deepest depth. So there is some uniqueness to the middle time point, and it'll be in the paper.
When you were comparing the Atlantic to the Pacific, is there any, concern for the difference in the euphotic zone depth?
Yeah. So when I averaged all of the particles, I grouped them into their depth zones based on their locations.
Okay.
So you're right, because the Pacific water columns, the euphotic zone was I can't remember an exact number, but it's 80 to 100 meters. It's a lot deeper, and it's more constant than what was observed in the Atlantic.
What was the significance of doing this during a bloom event, and how would that compare maybe to like a normal ocean moth conditions?
Yeah, so we were trying to just capture this high flux event, like a lot of export of organic material. So that was the big reasoning behind exports choosing those two field sites. Was this oligotrophic, high nutrient, low chlorophyll zone in the Northeast Pacific? It's low productivity, and then compare that to a very distinct difference in the Northeast Pacific or Atlantic during the spring bloom, where there should be a lot of export because there's a lot of production happening.
Any other questions? Hillary?
Is there any chemical approach that you think is a natural add-on or follow-up to what you did here?
Yeah, I think I kick myself every day for not analyzing carbohydrates in the sediment traps. That's a personal thing I have to live with, but I think lipids too. So that was a big thing. When I saw that high C to N ratio, I thought it would be carbohydrates because we were at the end of a bloom. Diatoms typically, when they're stressed, like there's not a lot of nutrients, they normally exude a lot of carbohydrates, which then makes things sticky and dense and everything sinks. But when I looked at the carbohydrates, there was a decrease with time. And then I was like, okay, where else could this carbon be coming from? So I looked at lipids, and there actually was an increase in the total lipids with time. So lipids could be playing an important role. But a great thing about lipids too is you can do a bunch of different sizes or different fractions, and they can be a little bit more specific to source. So I think lipids would be the natural next biochemical to go after.
Anyone else? I wonder, is there anyone on Zoom who has a question?
Kim, maybe? Kim, do you have a question? Oh, she only switched the camera off.
Great talk. I really enjoyed it. I will try and formulate and send you a question, but it was midway through and it split it out of my mind. But your talk was fantastic. It was a really great presentation of the data.
Thanks, Kim.
All right, well, let's thank Liz once more for her wonderful talk. There will be no Compass this Friday. I'm sorry, but you'll have to wait till next Wednesday for Hope Elliott to present on her graduate work.